Hi, welcome to the course on Quality Assurance Interview Preparation. In a tablet manufacturing facility, why positive pressure is maintained in processing area or service corridors. In tablet manufacturing facilities, pressure gradients are maintained to avoid cross-contamination of products through air. Usually processing areas are maintained under positive pressure with respect to service corridors. What is the purpose of stress testing in stability studies? Stress testing of the drug substance can help identify the likely degradation products, which can in turn help establish the degradation pathways and the intrinsic stability of the molecule, and validate the stability indicating power of the analytical procedures used. The nature of the stress testing will depend on the individual drug substance and the type of drug product involved. What is dead leg? A dead leg is defined as an area in a piping system where liquid can become stagnant and not be exchanged during flushing. Are forced degradation, stress testing, and accelerated stability testing same? Forced degradation and stress testing are not same. Stress testing is likely to be carried out on a single batch of the drug substance. The testing should include the effect of temperatures in 10 degrees Celsius increments, for example, 50 degrees Celsius, 60 degrees Celsius. Above that, for accelerated testing, humidity, for example, 75% relative humidity or greater, where appropriate, oxidation, and photolysis on the drug substance. The testing should also evaluate the susceptibility of the drug substance to hydrolysis across a wide range of pH values when in solution or suspension. Photostability testing should be an integral part of stress testing.
Why do we calibrate a qualified equipment instrument on definite intervals? An equipment or instrument can drift out of accuracy between the time of qualification and actual use. So it is recommended to calibrate and recalibrate the measuring devices and instruments on predetermined time intervals to gain confidence on the accuracy of the data. Why do we consider 3 consecutive runs, batches for process validation? Why not 2 or 4? The number of batches produced in a validation exercise should be sufficient to allow the normal extent of variation and trends to be established and to provide sufficient data for evaluation and reproducibility. First batch quality is accidental, coincidental. Second batch quality is regular, accidental. Third batch quality is validation, confirmation. Into batch we cannot assure the reproducibility of data. 4 batches can be taken, but the time and cost are involved. Explain about revalidation criteria of AHU system. AHU system shall be revalidated periodically as mentioned in the regulatory standards. AHU shall be revalidated in following cases also. When basic design of AHU is changed. When clean room volume is changed. When new equipment is installed. When a construction is carried out that calls for reconstruction of AHU system. What needs to be checked during AHU validation? During AHU validation, following tests shall be carried out. Filter efficiency test. Air velocity and number of air changes. Airflow pattern, visualization. Differential pressure, temperature and RH. Static condition area qualification. Dynamic condition qualification. Non-viable count. Microbial monitoring. Area recovery and power failure study. What is the difference between QC and QA? Provides the confidence that a product will fulfill the quality requirements, whereas QC determines and measures the product quality level.
What is the difference between GMP and cGMP? GMP means Good Manufacturing Practices. These are the standard guidelines given by Food and Drug Administration to make sure that a product is manufactured with safety and quality. cGMP, known as Current Good Manufacturing Practices, refers to recent and advanced updates to these standard guidelines. cGMP is up-to-date standard reference guidelines. Do you know any countries with names of their regulatory authorities? India, Central Drugs Standard Control Organization. USA, United States Food and Drug Administration. UK, Medicines and Healthcare Products Regulatory Agency. Japan, Ministry of Health, Labor and Welfare. Brazil, Anvisa. What is ICH and ICH guidelines? ICH is the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. It brings the regulatory authorities and pharmaceutical companies together to discuss the drug registration technical aspects. ICH provides guidelines on 4 aspects, that is quality, safety, efficacy and multidisciplinary guidelines. What is BMR and BPR? BMR, Batch Manufacturing Record, is prepared as a written file during the manufacturing a product by writing step-by-step manufacturing process and details about batch recorded here. BPR, Batch Packaging Record, is kept for every BMR. BPR depends on packaging operation. What are stability studies? Stability studies are necessary for developing the pharmaceutical products. It helps to evaluate the effect of environmental factors, for example, light, humidity, temperature, etc., on active pharmaceutical ingredient or pharmaceutical formulation.
What is the purpose uses of stability testing? Stability testing gives the confirmation as how the quality of study product changes with time. It finds out the study product shelf life, recommended storage conditions, as well as container closure system suitability. Also gives assurance to the patient regarding stability of a drug product. Stability testing is a regulatory requirement also. What is the time period required for long-term and accelerated stability studies? Long-term 12 months, accelerated studies 6 months.